Oh, that scared me.
I know, there we go. All right, so this slideshow today is about the cell cycle and about mitosis. So before we get started, let me switch that over there. Okay, so when we talk about the cell cycle, or before we talk about mitosis, we have to talk about the cell cycle. And the cell cycle and mitosis go together, although people spend a lot of time talking about mitosis and not so much the other part. So in this slideshow, here's what you're going to learn is we're going to talk a lot about mitosis, but we are going to mention the cell cycle here at the beginning. Even though we spend a lot of time talking about mitosis, it's only a very small part of the whole cell cycle, but it's a very important part. So what is this thing called the cell cycle? Well, in short, it's a repeated set of events in the life of a cell. The cell cycle is actually two big parts. There's interphase and then there's mitosis. Interphase itself is divided into three smaller phases, G1, S, and G2. And then mitosis follows the end of G2. The cycle itself, all of it together, is very highly regulated. If something goes wrong at certain parts of the cell cycle, sometimes the cell will trigger itself for a programmed cell death. And that programmed cell death is called apoptosis. This is a normal, healthy way to get rid of cells that are damaged. If the cell cycle is not regulated, you can have cells that overgrow or they grow too quickly. Cells that are out of control in how they divide, or if they're dividing when they are damaged, that can lead to cancer. So your immune system has some control over the cell cycle, but when that goes wrong, then you can have an unregulated process, and that can be bad. So what does the cell cycle look like? So here's a nice little diagram. Let me make it big and pretty. So here is the cell cycle. And the cell cycle is, like I mentioned, two parts. There's interphase and then there is mitosis. Mitosis itself, which a lot of people focus on because it's more exciting, is very small. It's only about this much of the cell cycle. Most of the cell cycle is interphase, and interphase is G1, S, and G2. And so what happens during G1, the first part of the cell cycle, is what we think of as normal cellular activities. The cell is growing in size. It's going to eat a lot. It's going to metabolize a lot because cells that are in G1, you can think of them like babies. They've just come into existence. So they're going to do everything that they need to do in order to grow. At the end of G1, okay, when they reach this part of the cell cycle, they've grown as big as they can get. There comes a point where cells cannot continue to grow bigger and efficiently get rid of waste and efficiently bring in food. So the only way they can manage still getting food and getting rid of waste efficiently is if they cut themselves in half or if they divide. So the signal to go from G1 over to the next part of the cell cycle, the S phase, the signal might be, scientists are pretty sure, that the cell reaches its limit, its size. When it gets to that limit, it does have to make a decision. Is it going to divide or not? Cells that do decide to divide will go on to the next stage, which is the S phase. Cells that don't divide, instead they do an alternate, they go into something called G0. And G0 is not a terrible place. In fact, some cells go into G0 anyway after they've reached their predetermined number of cell cycles. Cells that are in G0 are alive and they do everything to maintain your tissues. So, for example, nerve cells, muscle cells, they only divide once, maybe twice in their whole life. And once they reach their pre-programmed number of divisions, they go into G0 and they don't divide again. But cells that have to be replaced constantly, like your skin cells, they'll grow and they'll reach a point where they have to divide again and they'll go into the S phase. So cells that go into the S phase, they're in preparation to divide. The S phase is important because what happens during the S phase is DNA replication. In other words, every piece of DNA, every chromosome, if you will, a piece of DNA is usually referred to as a chromosome. It's a very definite piece of DNA, but that's what a chromosome is. So every chromosome, every DNA molecule in your cell is going to copy itself through that process that we talked about last time, where the chromosome divides in half and each half is used to build a new side. That happens in DNA synthesis or the S phase. After the S phase, then cells enter G2. If a cell enters G2, that's an important part of the cell cycle because cells that enter G2, let's see if I can write this word neatly. Cells that enter G2 are referred to as committed cells. What they are committed to is, oh, that looks kind of weird, but the word there is committed. Cells that enter G2, the reason they are called committed is because they will eventually divide. Whether they're good cells or whether they're bad cells, they are going to divide. So all along this route, there are proteins which check the progress of a cell as it goes from G1 through the S phase and into G2. And if a cell passes all of those checkpoints, it's allowed to go ahead and divide. So when you get to G2, there's not really any more checkpoints after G2. So that's why it's such an important phase, is that if a cell enters this, it's going to divide. The part of the cell cycle that gets the most attention is the mitotic phase. Historically, mitosis was studied because it's so much more elaborate under the microscope. We could see cells in any of the mitotic phases, but we couldn't really see a cell in G1, S, or G2. So historically, biologists believed that between one mitosis and the next, the cell was just resting because there was nothing visible going on under the microscope. But as our instruments became more sensitive, as the area of biochemistry advanced, and certainly cell biochemistry advanced, then scientists realized there's a lot going on in G1, S, and G2, even if we don't see it under the microscope. So mitosis really was quite well studied before we ever figured out what was happening in interphase. On the test, there's going to be a question, true or false? Interphase is a period of rest. The answer is false. The cell is not resting. This is where it's busy undergoing metabolism. It might be copying its DNA or it might be preparing for the cell divide. But interphase, it is not a period of rest. Interphase itself is made up of G1, S, and G2. So what happens if a cell finishes interphase, then what? Then it goes on to the mitotic stages. Oh boy, look at all these lovely, let me get rid of all these. I didn't realize they were going to stay on there on the next slide. Okay, there we go. So what's going to happen in mitosis? Well, you're going to divide out your genetic information. And when we use the word genetic information, we're talking about things that you pass on. A single chromosome contains many, many genes. A single chromosome contains many, many genes. And those genes themselves, they are the units of heredity. They're the ones that contain information about your physical traits. So many, many genes make up a chromosome. You have different numbers of chromosomes depending on what type of living thing you are. I'm human, you're human, I'm guessing. And so you have 46 chromosomes. That's your chromosome number. It is possible to have more or less than the normal chromosome number, but humans, the chromosome number is 46. We talk about them in pairs. And so the point of mitosis is to divide out those chromosome pairs. So how do we know about these chromosomes? Well, historically, it was a German anatomist who first used that word chromosome. His name was Walter Fleming. He studied cells under the microscope, and he applied dyes to what he was studying in order to better view what was going on under the microscope. The cells that he was viewing, they were actually dead. They weren't actually dividing actively. But when he applied the stain and they died, they were literally killed in the act of dividing. So it was almost like a snapshot of what's happening. And the word chromosome he applied to what he could see under the microscope because he noticed that the stains were picking up these thick strands inside of the cells. And so the word chromosome literally means colored bodies. He also noticed that at different points, he would see cells that had thick strands and uh, cells that had very thin strands. And so cells that were not actively dividing, he figured, or he reasoned, I should say, he reasoned that they were resting. Remember, he didn't know about interphase at this time, but he reasoned that they were resting. And so he said that those thin strands, he called them chromatin. But if they were thick, he called them chromosomes because he could view them in the act of them actually dividing. So from his notes, Walter Fleming was able to recreate a sequence of events that occurred during cell division, and he called this process mitosis. Now, mitosis itself is asexual. That means that even though it creates a new thing, that new thing is genetically identical to the parent cell. Mitosis doesn't need two separate parents. In fact, you have one parent cell that literally divides itself in half. And so asexual reproduction always results in cells that are genetically identical to each other and to the parent. There's no chromosome mixing, if you will. It's like making a cellular photocopy. So here are the steps of mitosis. Mitosis includes, and I'm going to skip down to the bottom here, mitosis has 4 stages: prophase, metaphase, anaphase, and telophase. So step 1 of prophase, and you know what? I'm going to stop right here. I'm going to stop my recording right here. That was the part that I wanted to record. I think I can stop my recording. There we go. I can stop my recording. All right, perfect. Not too bad. I liked it. Are you still with me, Amna?
Yes.
Perfect. Okay. That was just something I wanted to try and see how it came out on Zoom. I'd never actually recorded on Zoom, and somebody told me I could do that. I'm like, you know what? I'm going to try it on a tiny little part. Okay, good. I feel weird though when I'm talking. I feel like I'm not myself.
There's just a lot of background noise. That's why I muted myself.
Oh, no, no, that's fine. I was not worried at all. I knew you were still online. Okay, so here's the part we're going to draw out. So cell cycle has 2 parts. There's interphase, which itself is G1, S, and G2. And then there is mitosis. Remember that there is this G0, which is an alternate to G1.
I can't see your notes. I don't know if you're trying to show.
Oh, sorry. You can't see what I'm writing. How's that?
Okay.
Now you can see what I'm writing. And I'm looking up the page number here where you can find what's happening. It's also in the notes that I handed to you or that I gave you. But see page 125 of your textbook. as to what's happening in the G1, the S, and the G2 phase. Now, a description of what's happening in mitosis, that actually starts on page, I'm going to write this down, page 130 of the textbook. So if I go too fast, you can pick it up in page 130. So do you remember these terms? Prophase, metaphase, anaphase, and telophase. Remember this? Oh, perfect. Well, if you remember these, then this will be no problem. So these are the stages of mitosis. Remember that about 80 to 90% of the time, the cell is somewhere in interphase. That means only about 20 to 10% of the time is the cell somewhere in mitosis. Most of the time, your cell is in interphase. But when it gets to mitosis, this is when it looks exciting under the microscope. Cells that are in prophase, this is where we're going to have some profound changes. Your chromosomes are long, loose, and stringy. And one of the first events that happens in prophase is that chromatin, it's going to condense and organize into chromosomes. So your DNA is long, loose, and stringy like this, and then it starts to get tighter and tighter, and we use it to make a chromosome. It doesn't change the amount of DNA that you have, it simply changes the appearance of it. The next thing that's going to happen is outside of the nucleus, you're going to have these little fibers. They're going to be produced by these things called centrioles. So you have some centrioles that are outside of the nucleus. They're going to produce some fibers, and those fibers will attach to the chromosomes. So you start to see these little threads. Here's a centriole. And then here's another centriole. And they're going to attach these little fibers, which we call spindle fibers, to the chromosomes, because eventually we're going to use those fibers to move the chromosomes around. You'll start to see the nuclear envelope disappear. It's going to disappear because we're going to move the chromosomes around. And so we need all of the available space that we can use inside of the cell to do that. So a cartoon diagram of this event, if I can show you a cartoon, let me know when it's okay to switch over to my PowerPoint.
Oh, you're good.
Okay. So a cartoon, kind of a representation of this happening, is right here. Okay, so this is a cell that is here in the middle of prophase. This right here is one spindle. Right here is the other spindle. It's actually centrioles. The spindle is the centrioles plus the threads. So this is the spindle fibers. So the centrioles have produced these fibers that have attached to the chromosomes. The fibers are going to be what we use to move the chromosomes around. In order to best move the chromosomes around, these centrioles are going to be completely opposite of one another. They start out close to each other, and then one of them moves to the opposite side as they're producing fibers, as they're attaching it to the chromosomes. Meanwhile, the chromosomes, they used to be long, loose, and stringy, and now they're getting nice and thick. At the end of this process, the nuclear membrane is going to dissolve so that we have lots of room to spread those cells around. Here's another diagram. Actually, this is an actual cell that's going from prophase that's going into prophase. So here are two cells. One's an animal and one's a plant cell. They are in interphase, so they are not dividing, but they're getting ready to divide. I can take my tracker here and I can trace right the edge. Oops. Okay, I can trace right here. This is the edge of the nucleus. That's nuclear membrane. And the same thing here. That's the nuclear membrane. And the cell membrane appears red because the dye is all spread out because the DNA is spread out. When it goes into prophase, then you start to see the threads form because the chromosomes are thickening. It's real dramatic inside of a plant cell. The nuclear envelope is starting to disrupt. It's starting to disrupt. In this picture of the animal cell, it's not quite as disrupted as it is here in the plant cell. So that's prophase. In metaphase, this is pretty dramatic. The chromosomes have been organized in the middle of the cell. I always say they look like messy stitches in a plant. Here's the chromosomes here. These red looking lines, that's what you can see of the spindle fibers. They're still there. And then right there in the middle, that is where the centriole is. One thing interesting about plants is they don't have centrioles. They have fibers, but the fibers are actually attached to the cell wall. And it's the cell wall that's actually causing the movement of the chromosomes. But the idea is the same. Whoever's moving the chromosomes, they're going to move them so that they eventually get to the middle of the cell. Okay, so let's write that down before we forget all that wonderful information. So what makes metaphase metaphase is that the chromosomes have been directed to the middle of the cell. And the middle of the cell is referred to as the equatorial plane. You can think of it like the equator of the Earth, okay? The equatorial plane. How cells know where this is, that's a bit of a mystery. Scientists have not figured out how a cell knows where its own middle is. That's crazy. They don't know, but cells know. They seem to know where the middle is. And they direct their chromosomes, and those chromosomes line up perfectly in the middle of the cell. It's actually right here, the middle of the chromosomes, that aligns in the middle, and they form what are called the metaphase plate. So when you see chromosomes lined up in the middle of the cell, they form what's called the metaphase plate, and they form it along the equatorial plane. That metaphase plate, where the chromosomes are lined up, okay, the arms may not be perfectly lined up, but the middle of the chromosome is lined up. That's one of the straightest lines in nature. Nature does not have a lot of straight lines, but how cells know where the middle is and how they know to line up their chromosomes almost perfectly straight, that's one of the coolest, most mysterious things I think about cell biology. How they know and how they can make them perfectly lined up in the middle. Better than any company front that you'll see at any military school except NIMI. We have the best company front, right?
Yes.
tell, tell me who's in formation. I'll tell you how good of a company front it is. All right, anaphase. What's going to happen next is that these fibers that we've used to assemble the chromosomes in the middle, they're going to shorten. So all of a sudden, the fibers are going to shorten in opposite directions. And when they do that, they're literally going to pull the chromosomes apart in half. So the spindle fibers shorten, and they pull chromosomes apart. And those chromosomes, they move to opposite poles. They move to opposite poles. Sometimes you'll see that they use the word chromatids when they're describing this. They'll use the word chromatid. And I'll show you why. When you have a chromosome that looks like a puffy letter X, okay, this side is called a sister, and this side is called a sister chromatid. So when it looks like a puffy letter X, that whole thing is called a duplicated chromosome. That's because after DNA replication, you have two halves. Before DNA replication, you just have one side. But when you go through DNA replication, you copy that side, and now you have the original and its copy. And I'm just drawing one side dark and one side light to show that the original versus its copy. So if it's a puffy letter X, it's referred to as a duplicated chromosome, but the two sides are exactly the same. So when you divide them in half here in anaphase, you're not actually getting fewer chromosomes. You're simply separating duplicates. So half of the information goes this way, half of the information goes that way, but the amount of information is considered to remain the same. That process of them separating continues until those sister chromatids are completely separated from each other. I mean, they're completely separated from each other. And when they're completely separated from each other, then we say they are in the middle telophase. Excuse me, Johnny's being crazy.
It's okay.
Sorry, Amna, my neighbors are getting home after a long day at work, and my dogs like to bark. Okay, I had to distribute some snacks so everyone would be quiet. All right. So you're looking at my paper, right?
Yes.
Okay. I misspelled chromatids. There we go, chromatids. All right. So telophase, the cells in telophase when these little fibers has moved as far apart as they can go. So those chromatids, each set, if you would, the sets of chromatids have moved as far apart as they can. And now we're going to do prophase in reverse. So the nuclear envelope is going to reform, but around each set, the spindle fibers, the threads, they're going to fall off. Okay, they're going to detach and they're going to dissolve along with the centrioles. So the fibers detach, they go away, and the separated sets of DNA, you're going to form a new envelope around each one of them. Technically, this is the end of mitosis. Okay, technically, it's the end of mitosis because mitosis is really just dividing DNA. That's all really mitosis really is dividing the DNA. You still have to divide everything else. So the last part of telophase includes something called cytokinesis. This is dividing the cytoplasm. So mitosis divides the nucleus, but cytokinesis divides everything else. In an animal cell, you have an inward pinching. That inward pinching is referred to as cleavage. You can also call it a furrow. So animal cells, this is how they divide the cytoplasm, is that the cell membrane literally pinches off in between the two separated sets of DNA. So not only does the cell know where the middle is, it knows where to do this inward pinching so that it eventually separates into two cells. In plants, this is accomplished a little bit differently. There's no inward pinching. Okay, plant cells are usually represented as kind of boxy because they have the outer cell wall, and then they have the cell membrane. So here's the nucleus, and here's the other nucleus that is newly formed. The way that we divide the rest of the plant cell is we have something that starts forming right in between them that's kind of like a baby cell wall. That's referred to as the cell plate. And the cell plate will grow and develop over time to eventually become the new cell wall. And when it becomes a new cell wall, that is what divides the rest of the cell. I will tell you that that is a test question, is how does cytokinesis differ in animals versus plants? Right, that is a huge, that you'll see that on the test. Do you have any questions for me on them?
No.
Yeah. So again, the pages that I would refer you to if you need some reinforcement, pages 125 and pages 130 of your textbook. I have one homework assignment left for you. Okay. I have one homework assignment left for you, and it is an ed puzzle.
However,
It's an ed puzzle. And so the ed puzzle is a video over the cell cycle and mitosis. It is a, let me look and see here. Yeah, it's available. So it is available. That is your last regular assignment of this semester. Yay! There are some things that you still need to do for me. Remember
right.
those assignments, okay? Have you been working on them?
Yes.
Okay, the assignments are not due till Thursday, but what's due tomorrow?
The test. We have the
Right.
test tomorrow, and then I have to look in Canvas. I think it's just a test. Yeah, the
Yeah.
makeup.
So tomorrow's the makeup test. And then Thursday, the assignments are due. You can turn them in earlier. And then Friday is the final exam.
All right.
So tomorrow, I will be available online after we'll meet for the test, and then I'll be available, but there's nothing else that you need to do for me that's new. All you have to do is the makeup work. So tonight you have an assignment over, let me see here. Oh, all you have is the ed puzzle because the other one we did in
Right.
class together. I'm going to give you full credit for that. So all you have to do for me tonight is the ed puzzle. Tomorrow we'll take the test. Thursday we won't meet on them. My husband is having a medical procedure
Right.
on Thursday. You can
Are we
text
meeting
me.
later?
We
Or
can meet later.
okay.
If you want to meet with me online, we can meet later, like at 5 in the evening.
Yeah.
But
Oh no, we don't meet Thursday, right? That's
yeah,
just
we're not scheduled to
okay.
meet Thursday. If
Right.
you want to meet with me, you can, but it won't be until 5 in the evening.
Okay.
But you can text me on Zoom all day long. Okay.
Yes, ma'am.
What time would you like to meet tomorrow?
Our normal time 1.
At 1 o'clock? Okay, I will be here at 1 o'clock. If something changes, just reach out to me, okay?
Wait.
So hang on, you're almost done. Amna, you worked so very hard. You don't know, maybe you do. You don't know how much I want you to succeed this summer, so you don't have to take biology anymore.
Yes, ma'am.
You probably want to succeed as much as I want you to. So
Yes.
I'm going to send you a copy of these notes. I'm going to post them to Canvas, okay?
Okay.
And I will see you tomorrow.
I'll see you tomorrow, ma'am. Bye bye.
Bye-bye.